Thank you for giving this opportunity. So I'd like to, focus on our recent focus in our lab about a 3D culture platform, which we call it actually Rapid Rare Cell 3D Expansion, for short for RCE. Okay. So why we're interested in this one? Okay, first of all, okay, we all know that animal studies, there's already a global consensus. When you conduct an animal study, you have to consider the 3R, the so-called replacement, reduction, refinement, right? And there's even global consensus. We should eliminate animal testing altogether by 2035, okay? So doing this is not just because of the human reason, okay? So just read this quote here by Dr. Richard Klausner, back to 1998, okay? Some of you may have seen this before and probably also feel amusing, just like when I saw it. But I think it's supposed, seems funny at first, but it's also speak of the truth, which is that a lot of time we're doing the medical research, but really just, just try to cure mouse. And we all know that ineffective models result in high failing rate. So 9 out of 10 drugs cannot even go into the clinical trial, okay? So effectiveness is also very important. So animal is not an option. What kind of model is better, okay? As a surrogate, better than 3D, better than animal model, okay? So there's a lot of excellent work in the past few years, and we actually, as a whole, the global community made a lot of progress, okay, including the folks in our Stanford community. So showing that 3D model, 3D cell culture, okay, that resemble the morphology and the function of the actual system might be the solution, okay? Why this is the case? So here I actually want to show you 2 video clips recorded in our lab to give you some taste, what the difference between the 2D culture versus 3D culture, okay? So both systems have the core blood natural killer cell, the immune cells, the small little particles here, okay? And they try to kill cancer cells. But here's a twist. On the left is the cancer cells on the tissue culture plate forming a 2D structures. But on the right is the cancer cells forming spheroids on our RCE plate, okay, which I will talk more about its mechanism and other application. But here, just bear with me. That's a spheroids that are grown on our RCE plate, okay? So now let's see what happened here, okay? So the right, left, you see that, like natural killer cells start to invade cancer cells. And then the cancer cells are pretty defenseless. And, in no time, so within the 46 hours, you see that pretty much the cancer cells were killed and NK cell declared the victory, okay? But the story could be quite reverse, okay? Now you see that now if the cancer cells band together as a 3D structures and the natural killer cells almost cannot find a room to attack them. So in the beginning, then you see like there's an intact spheroids. And please pay attention to all these large spheroids and the 2 spheroids here. The time goes by, these cells even like merge together, forming a large chunk of 3D structures. So at the end of 46 hours, like let me quickly fast forward here, then you see that, they pretty much all the 3D spheroids are intact and maintain a very smooth morphology. So now the question to ask you, okay, if you're researchers that study the NK cell effect, especially for the current, the modern area of immunotherapy, okay, especially for the autologous immunotherapy, which data would you believe? So is the NK cell effective or not, okay? So the answer could be quite opposite if you use a different cell model. Okay. So 3D culture, then I hope that I should show you that 3D culture is quite important. So everybody has a different angle to cut into this area. But in our lab, our approach is really from the material designs. Okay. So first we synthesize and integrate cross-dimensionally. What I mean by that is that first we care about the nanostructure of the surface. So how the polymers, how the biomolecules and so forth integrate together, okay? That kind of molecular architecture, then we can use the engineering approach, such as photolithography, 3D printing, and so forth, to forming the micro-scaled precision. And at the end, depends on what kind of biological or medicinal application that we integrated to a device level. In this case, we actually integrated into a microfluidic chip. So as an example, like for the circulating tumor cell isolation, okay, from the blood. And this actually is, like one example that we spin off a company called CellMax Life, which is located in Sunnyvale right now. And while I'm preparing this talk, and then I actually see also Dr. Shai Friedland's recent work in ASCO, and using the CellMax fluidic chip, to study the early detection and the prevention of colorectal cancer. So this is the working flow in our lab. So from the surface, from the materials point of view. So this is another example of what we can do in our lab, which is basically reduce the extracellular microenvironment into layer by layer structure. In this case, and then you see that we can first form lipid bilayers on the surface, and with one layer of the protein and the other layer of the protein, and forming a complex extracellular surface. And of course, the substrate itself can be quite flexible in a way that it could be glass or it could be a polystyrene plate, which is typical for the tissue culture plate design. Okay. So in this way, then you can see how we can control the cell morphology from the 2D to 2.5D and even to the 3D spheroids. All these things simply just controlled by the surface properties, nothing else, nothing more, okay? And in particular, today I'm talking about RCE. This is a 3D cultural platform, okay? So what is RCE? Okay. So RCE, in summary, is a robust 3D cultural platform made of synthetic biocompatible polymer coating. Why is a synthetic polymers? Is obviously, because it's not an animal-derived component, so it's easy QC. And eventually it could be clinically applied for various settings. And also they can be coated on different kind of cultural dish configurations, which means this is very important for the user is that you do not need to change your current equipment or setting, okay? And you can use RCE as easy as using any 2D cultural method. So by using the RCE coated 96 wells, we can actually generate the RCE tumoroids. In this case, using the colorectal cancer cell line HCT116. After 5 days of culture, and we have this very straight lines showing you that, okay, the spheroids counts per wells are quite consistent across the different wells, as well as the spheroid size are fairly consistent. So for the initial seeding of 300 cells, after 5-day culture, we also get on average 300 spheroids. And in each spheroid, we have about 400 to 1,000 cells. So why this is such a high yield, extremely consistent, and also the spheroids are so uniform? And why is that? So I want to show you the video clip to show you single cells on RCE plate. So it actually, okay, can derive the single cell proliferation okay, purely from one cells derived to the a spheroid on the RCE plate. So that's why when we have the 300 seedings, and then we still receive the 300 spheroids at the end of the day. Okay. So this kind of single cell derived organoids is important in that if you compare with the conventional 3D culture platforms, which generates work by the cell-cell interaction, meaning that you need to have a lot of cell density to begin with in order to form the cell aggregate at the end. But the RCE can start from one cell, okay? So other than you have a high yield of spheroids at the end, but also it's ideal for low abundance cells expansion. For example, like stem cells, circulating tumor cells, or even needle biopsy tissue and so forth. You can use this to expand them. Okay, just to summarize what we have done so far. So we already demonstrate that circulating tumor cells, tissue, stem cell, or cancer cell lines can all form RC3D cultures. Okay, so how these then can connect to personalized medicine? So I'd like to show you that what we envision the personalized medicine, perhaps the prerequisite is that then you need to be able to diagnose one person systematically, regularly, okay, and preferably using non-invasive way, such as drawing the blood or needle biopsy and so forth. So this kind of sample specimens flexibility is probably the prerequisite for somebody to achieve personalized detection. Okay, so our approach is that, okay, once you have this specimen, okay, maybe the cells are really low abundant, but we can first expand it in a RCE culture. Okay, after that, the multi-omic analysis, including the immunostaining, okay, or NGS sequencing, or last but not least, is a direct cell testing on drug can be achieved. And of course, if we want to make it clinically relevant, we want to make sure that, okay, everything has to be done within a month. Okay, otherwise you cannot plug into the clinical flow. So if we backtrack that, and then we know that, okay, 2 to 3 weeks of culture time to generate a sufficient amount of cells for the subsequent study become very important. Okay, so can we do that? So I want to show you some data here. Okay, this is the fresh tumor tissue derived RCE spheroids. So it's basically the colorectal cancer or liver cancers, very kinds received from the hospitals. And this is a breast cancer. As you can see that all the primary tissue, fresh tissues, and after 2 weeks or 3 weeks, or even less than about 1 week, then we already have a good amount of expansion and with distinctive different morphology. And as a control, for example, this breast cancer case, within 3 days, and we see the spheroid form on RCE plate. But as opposed to that, there's nothing on the TCP other than some cell debris. So this actually shows some good, successful results from RCE. And obviously, one person will ask, okay, if you have that 3D spheroids, are they actually tumor cells, or you actually just grow some fibroblast, for example? So this is, animal studies and xenograft to show you that this is liver cancer patient on stage 2, resected cancer tissue expanding after RCE. And then we do the inoculation after 8 weeks, and then we get quite a large tumors here. So this is one proof. And then the other one is to show you another example is a colorectal cancer surgical removed tissue. 2 weeks, we see the spheroid form. 4 weeks continue to grow even larger. But at 2 weeks, this is our clinical time point, deciding point. So we want to use this sample to do the NGS sequencing to identify the mutation hotspot. And quite interesting, you can see that the functional mutation identified after 2 weeks of RCE derived actually a lot more than the original tumor tissue. So this is actually telling you that, okay, first of all, that means that we indeed enrich the tumor content. And secondly, it's actually help you to identify more mutation than the original tumor, which has lower tumor content. Okay, and also the other important thing is that then to just give you the more mutation, then actually then you probably can take more actions clinically. Okay, what about circulating tumor cells? Okay. Here show you some examples. This is colorectal cancer, breast cancer, head and neck cancer. So 2 days you don't see much, okay, but then within 2 weeks you start to see the spheroids grow, and then within a month, and then it's even grow larger, the same as breast cancer and head and neck cancers. So similarly, then we can also have the breast cancer, so 2 weeks, 3 weeks, or this also 2 weeks, and this is head and neck cancers. All can grow to large tumoroids after 2 weeks a period of time. And then we can also identify their tumor contents using the NGS DNA sequencing. So again, we also detect a lot more functional mutation than the original tumor tissue, okay, from the same patient.
Hi, Ying. So sorry, but we be able to wrap up in about a minute, just to stay on time.
Okay, all right. So so the last, okay, so I'll just say, I would just want to show you the Dr. Pingren's, Mark Pingren's study. So he said, okay, so with all these merits, what about let's just directly apply the chemo drug on this CTC-derived tumoroids, because otherwise, these chemotherapies, and there's no other way for us to identify the right one for the patient. So here to show you that indeed we can do this, on the various chemo drug and show the resistant or susceptible for individual patient, and we can generate cutoff out of this cohort. Here, and we show you that even this study cut short because of the pandemic, but with this limited set of data, we show all the clinical concordance is actually 100%. Okay, and this is some continuous study in gastric, bladder, and also non-small cell lung cancer. Okay, with that, and then I'm just conclude, and I'd like to thank my group members in both in Taiwan Genomic Research Center, as well as the Stanford in the Chemical Engineering Department, especially in the middle is my long-term mentor and the collaborator, Professor Kurt Frank. And many of us are right now are in Taiwan, but we're looking forward to returning to campus soon, okay? And here, and then I'd like to thank all our PIs who conduct the clinical research and also the design for us. So thank you very much.